Okay. All right, good morning, everyone. Let's get started. And then it will be sooner that we'll reach the table of full of food tomorrow. So just a reminder, there's no lab today for those of you who are in Wednesday lab. Thursday, Friday, obviously no lab. The school is closed. And I have posted the study guide, if you haven't already seen it, for our next exam, which is a week from today. Also, I'm going to hand back your quiz from last week today at the end of lecture. And so I'm going to start by finishing up our discussion of epidemiology, and then we'll move on to pathogenesis of disease. So I think we left off with discussing the groups that are in place that are part of this network of epidemiologists that help to achieve the goals of controlling the spread of infectious disease, preventing infectious diseases, and designing effective treatments and recommendations for effective treatments of infectious diseases. And the major public health institutions go all the way from the local level, where each of our towns will have a public health department, to the national and international level with the CDC and the World Health Organization. And other countries have their counterpart version of these public health institutions in their societies. And so the World Health Organization really works with these other national organizations from other developed countries to achieve these goals. So one example, I think did I already go over this example or not yet? Okay. So one example of the success of vaccination, which is one of the major goals of public health as far as preventing the spread of disease and controlling a disease and also preventing the incidence of a disease, includes, there are many examples of success, but one of them that is shown here in a graphical form is the effect that the diphtheria vaccine had on the incidence of diphtheria. So here, remember, the term for morbidity measures what? The number of individuals infected out of a population that's susceptible, right? So morbidity would be the number of people getting sick. The diphtheria vaccine was used in the 1950s. And so when you look at the morbidity or the number of people getting sick within a segment of population, so now they're only looking at 100,000 people as a group, and you look at that across the population of different ages, you can see that the older individuals who are not receiving this vaccine as a child have a much higher rate of disease, okay, than those that are younger than the age when they started to implement diphtheria toxin as a routine childhood vaccination. Okay, so there's a much lower morbidity in the case of those that were protected by the vaccine. And that's the goal that epidemiologists like to see achieved with the recommendations for vaccination for any infectious disease. And one important thing to note about why this vaccine may have been so successful is the nature of the infection. So Corynebacterium diphtheria, the bacteria that causes this illness that affects the nervous system, the heart and the kidney in in the most severe cases. Otherwise, it's a respiratory disease that's acquired by inhalation and spread by respiratory mucus and secretions, also in respiratory droplets. The immunization, you can see, is effective at preventing disease. Those who do get infected will get antibiotic and antitoxin treatment because of this toxin that mediates the damaging symptoms, the most severe symptoms, the most severe damage to our bodies, our nervous system, our heart and kidney. So it's really this type of infection, whether it's bacterial or viral, that is passed from human to human without an animal or an environmental reservoir, that we see the best success as far as reducing, controlling disease, preventing disease. And I already mentioned smallpox as an example of one infectious disease caused by the smallpox virus, vaccinia virus, that has been eradicated completely. It's no longer in the population because successful vaccination that started in the 1950s and was ended around 1979 when the World Health Organization declared that there were no more cases of smallpox in the world. And so without that human reservoir, the human host could not get this disease. Before that, and the reason that smallpox was chosen as one of these diseases to focus on as far as prevention, there was a very high mortality rate. So mortality is the number of people who die from the disease. And if you look at that mortality rate in the general population, it was around 30%. But when you look at it in age 5 and under in children, it was 80%. So it was really, in general, infectious diseases tend to have higher mortality rates in the very young and the very old who tend to be immunocompromised. When we talked about vaccination, we talked about how originally for centuries this practice of taking the pus from one infected person and using it to inoculate another susceptible person was used. So that's variolation. But in this effort mounted by the WHO, it was a cowpox vaccine, so a similar virus that was found to elicit the same similar enough memory or adaptive immunity, this long-term immunity, that could cross-react with the smallpox virus. And that vaccine was successful at protecting against smallpox enough so that when the efforts to vaccinate populations worldwide was undertaken, it was successful. So this took many years. So remember, they started in the 1950s when there were still 50 million cases worldwide, and it took a good almost 30 years to succeed in eradicating, and it meant very good public health records of where the disease, the incidence of the disease occurred. Also, efforts to distribute the vaccine to enough people so that those that didn't get the vaccine could be protected by what kind of immunity? Herd immunity. So this protection that when most of the population is vaccinated, they're not going to be a reservoir for the infectious agent or the microorganism that's the pathogen. And in that way, the program was successful. So now we don't even get the smallpox vaccine because it's actually, as a side effect in rare cases, will give more severe symptoms than the chance of catching smallpox from the general population, since there's no reservoir for it. So we don't get that vaccine anymore. And the only place that smallpox virus is kept is in research in a couple of research laboratories around the world, only 2 to my knowledge. And there's an ongoing debate about whether to preserve this virus for the purposes of research and study, or whether to destroy it completely and eliminate it from our world environment. Yes?
Isn't that at this point sort of academic since they've already sequenced it?
They've sequenced it, and but there are still studies that are ongoing. Even this year, there was one study that was published in a scientific journal. I don't remember which one. But there's still research done on the smallpox virus.
Oh, no, I mean, it's academic whether it's destroyed or not, since
Oh, yeah.
you find the record of what
Is
the
that-
genetic code is and you create it, at least theoretically.
Oh, I see. Yeah, I don't think we've gotten to the point where we can recreate a virus just from the sequence. You could probably use another virus and genetically manipulate it to be very similar to smallpox, but yeah, I don't know. It's mostly an academic argument, but there is this concern that if the virus got into that, a virus that was not attenuated in any way got into the hands of the wrong group, it could be used as a bioterrorism agent. Okay, so that was one example of success. Now, remember that smallpox is one of those infections that has humans as a reservoir and humans as a host. And we know that the virus is the pathogen or pathogenic agent. If we look at other infections like rabies, what are we looking at that's more complex? It's still a virus, that's the infectious agent, still a human host as far as for the disease. But what's the other variable in this story as far as the reservoir? Animals become a reservoir. So now the efforts to control and prevent this disease become much more complicated because you have another organism, another population of organisms in which you need to be concerned. So it's much less likely that an infection like rabies would ever be eradicated versus other infections like polio that still just have a human host and a human reservoir are much more likely to be eradicated via vaccination efforts. So what about emerging and re-emerging disease? So there's several variables that contribute to the emergence of a new disease, expansion of our populations into areas, geographic areas that were not populated before. So coming in, disrupting the habitats of animals that we might not have come in close contact with before, and then sometimes that infectious agent can cross the species barrier. Also just changes in these microorganisms over time. They become more or less virulent. If there's a breakdown in the public health system due to a natural disaster, we can have outbreaks or reemergence of disease. So a good example is after the earthquake in Haiti, there was an outbreak and an epidemic developed of cholera. Cholera has that reservoir of water, so it was contaminated water. So having these public health systems of safe drinking water and separating the wastewater from the drinking water supply are really crucial to protecting us from many infectious diseases. Also, any kind of disruption, whether it's a natural disaster or war, that would contribute to the breakdown of these infrastructures and these protections that we have. And climate changes mean that some infectious diseases that were not seen in areas of the world are now spreading to new areas, so emerging in new, unexpected areas. So one example is every summer now, the public health department goes out and collects mosquitoes and looks for what virus? West Nile virus. So West Nile virus, as its name implies, started much farther south and also east from here. But this infection that can cause encephalitis in the most severe cases is now a reality here in New England every summer because of that mosquito population that is a reservoir for and the bird population that's a reservoir for the virus. Okay, so any questions about epidemiology before I go on? Okay, so now I'm going to talk about pathogenesis. And from chapter 15, I want you to remember the 4 patterns of disease that most pathogens are required to take in order to cause infection. Remember that infection is different from disease. Infection is the colonization of the host by a pathogen. And remember that many individuals will be asymptomatic carriers of a pathogen. They won't have any damage, any symptoms that come with an infection. In the cases of infections like cholera, 95% of the people will get infected, but they won't get any disease. But those that do get disease are the ones that experience the symptoms. And disease means that it's infection with damaging effects. Damage is manifested as the symptoms that we feel. And the damage can be due to pathogenic products, toxins, or it can be due to our own immune response. So inflammation, and in the case of inflammation and fever and these other responses we have in order to eliminate the pathogen. So, but the basic difference between a pathogen, which is this relationship, this parasitic relationship that a microorganism will have with us in that it causes damage, and those microorganisms that are non-pathogenic, is that of virulence. So pathogens have virulence. They have the capability of causing damage. They may have low virulence, in which case they have caused very little damage and mild symptoms and not life-threatening, or they may have high virulence. This is a measurable characteristic of pathogens, and it's really a measure of the products that are encoded by their genome. So their DNA is fundamentally different from a non-pathogenic organism. Whether it's a plasmid that carries virulence factors that they've acquired from another bacterium through some kind of horizontal gene transfer, or whether it's a phage conversion, lysogenic conversion, something acquired, a toxin, for example, acquired due to bacteriophage incorporating into the genome of the bacteria. But there are many ways that these traits, these virulence factors, or the genes encoding the virulence factors can be acquired. And it's the the quantity or the degree of damage which these virulence factors can cause in us as a host that can be measured and assigned as a highly virulent organism, pathogen, or less virulent pathogens. For the most part, though, these 4 steps that are required for disease of attachment, infection, or colonization, and invasion and toxin production, whether it's before or after invasion, these require special traits that are part of these virulence factors. So things like adhesins that are proteins that are encoded by pathogens that enable them to attach to our tissues in a specific and strong way. Fimbriae are also an appendage that some bacteria have, others don't, and capsule. They both mediate attachment. Colonization, then, enzymes are often helpful in mediating colonization because they break down the extracellular matrix of the host tissues. Hyaluronidase is one example. Invading the barriers, so there are many different ways that pathogens get across these protective barriers of our innate first line of defense of the innate immune response. Some of them are directly injected into the blood by a vector like a mosquito, whereas others are able to penetrate the tissue because they have motility, like Helicobacter pylori being able to pass through the mucous membrane, or like we'll talk about Shigella and how it utilizes actually our immune surveillance in the gut to get across the epithelial cell layer lining the gut and the mucous membrane there. In general, another set of virulence factors that will be associated with pathogens is their ability to evade our immune response. So whether they have a capsule that's kind of like this cloak of invisibility that protects it from being recognized by antibodies, or whether they produce some enzyme that will degrade a key factor that's important for chemotaxis, for example, complement proteins, or whether it's able to avoid phagocytic cells, or once inside the phagocytic cell, able to escape from the phagosome before the phagolysosome forms. These are all virulence traits that are associated with pathogens that non-pathogenic bacteria will not have. They would be readily destroyed, either tagged by complement or antibody and destroyed by phagocytosis. And one of the most common ways that pathogens cause damage, especially those more virulent pathogens, is by the production of toxins. There are 2 major classes of toxins. There are exotoxins, those products that are produced and secreted by a pathogen, and there are endotoxins, which is LPS. LPS being a portion of what part of the gram-negative bacteria? Sorry? The outer membrane. So outside of that peptidoglycan layer, there's an outer membrane that's made up of a lipid bilayer. Part of it is phospholipid and part of it is lipopolysaccharide. Now, in the polysaccharide portion of it, we have things like the O antigen, which is recognized by our immune response. So we have antibodies that will recognize the different O polysaccharides in the outer membrane. But it also contains a lipid portion called lipid A, which is the toxic portion. And because it's an inherent part of the structure of the gram-negative bacteria, it's endotoxin, so it's not secreted. It's a part of the structure of the bacteria that can be toxic. Even when the bacteria are destroyed by antibiotics, the lipid A is still intact and still can have its toxic effects, which is why septicemia caused by gram-negatives is so life-threatening, 50% mortality rate in even when treated. And remember that lipids make a very poor antigen, so we don't have protection against the lipid A by antibodies. We have no antitoxin to use to treat this toxemia. Finally, if I have time, I'll touch upon how viruses damage and evade cause damage. So we know already from their life cycles, if they lyse the cell to release the virions that have developed in the cycle of replication, then they're going to cause damage to our tissues. But they also have means of evading recognition by the immune system. And that means either outside the cell evading antibody. So remember, antibody, once it binds to the virus, it's neutralized or ineffective at entering a host cell. Or when it's inside a cell, cytotoxic T cells will destroy that, will recognize that that cell of our own tissue is infected and destroy it by secreting enzymes that create holes in the surface, in the plasma membrane, and deliver another set of enzymes, granzymes, that will induce programmed cell death of that cell. So infectious diseases, we've already talked about epidemiology. Pathology is the study of disease. Etiology, already went over as far as Koch's postulates. But today I'm going to focus on pathogenesis, which includes infection, which is colonization by a pathogen, and disease, which are the damage caused. So all pathogens, by definition, have the capability of causing damage. That's the definition of pathogen and what distinguishes it from microorganisms in general. Now, there are a number of ways that human pathogens can enter. We already talked about the relationship between entry and exit. That includes entering any of the mucous membranes, the skin. The perinternal route would be some mechanism of delivery directly past these barriers. But a pathogen will never enter directly into our blood unless it's through this perinternal route, has some means of some carrier that delivers it into our blood. It will never enter directly into our liver or our spleen or our heart. These are not portals of entry for pathogens, although they may eventually end up in those tissues and cause damage there. We talked about the importance of infectious dose and that being a variable in as far as the virulence. Adherence is also these various traits, virulence factors that some bacteria have and others don't. The more easily they can adhere to host tissue, the more effective they are at the next steps of infection, colonization, and invasion. The step of penetration or evasion of the host defenses includes protection of the capsule, this polysaccharide layer beyond the cell wall. Some enzymes that are secreted, like hyaluronidase, which degrades the hyaluronic acid in our tissues to help the pathogen penetrate these tissues. Also, simply evading the antibody response by constantly changing its appearance and changing its disguise by, for example, shedding its pili and expressing a new set of pili that are different from the original, have different antigens on them. So the original antibody response would be effective against a portion of the population that hadn't expressed a new set of antigens, whereas this portion of the population that will be able to persist will then be able to grow up and become the majority of the population. So it's always this race between our relationship as far as our host immune response mechanisms that want to eliminate the pathogen and the ability of the pathogen to evade host defenses. Finally, there are some products that can affect our own tissues. So siderophores are molecules that bacterial pathogens will produce to acquire iron. That means it's taking iron away from our own cells and tissues that also need it. So we're competing for this important resource. Damage by toxins, whether they're endotoxins or exotoxins. The lysogenic conversion also is related to toxin production. And some have enzymes that will cause the lysis of cells. So these cytopathic effects. Most of the damage is due to toxins. And we'll talk about the different types of toxins and how they mediate damage. But basically, it's this balance between our own barriers and innate and adaptive immunity versus the virulence determinants that a pathogen has. And the four key areas where pathogens will have additional traits beyond what a non-pathogenic microorganism or microbial agent has will include those traits that enable it to be transmitted, so how long it can survive outside of a host, the attachment and invasion, crossing these barriers that we have. Motility is often a key factor in this ability to invade our tissues. And then the infection or colonization as well as disease. So the number, the dose required to cause these symptoms. The patterns of disease include toxin production. So some pathogens don't have to colonize or infect us in order to cause damage. And that's things like foodborne intoxication, where Staphylococcus or Clostridium botulinum are secreting toxins, and it's really only the toxin that has to survive the stomach acid in order to have its damaging effects of causing food poisoning. But in most cases, pathogens have to colonize, so they have to attach and then multiply in large numbers before the toxin production and the damage will ensue. So examples of that are Vibrio cholera and E. coli, these gastrointestinal infections, diarrheal diseases, that once they colonize and produce the toxin, they'll have this effect of imbalancing the electrolyte and the water secretion in the gut, in the colon. And others need to invade the host tissue, usually to find the resources they need to grow, as well as these are ways that they evade the host immune response. Some of them will cause disease without toxin production, just by invasion of our tissues. And that includes Mycobacterium tuberculosis, forming these tubercle growths in our lungs. Also, there are others that invade and then produce toxins. And that's the difference between, say, a Streptococcus pyogenes that's causing strep throat, which doesn't invade, it colonizes our throat, and that causes the flesh-eating bacterial infection that does penetrate that barrier of our skin, and with the toxin production cause a lot of damage So remember that virulence is encoded by DNA, so part of the genome, and these factors are then expressed as proteins or production of toxins. And so it's the virulence determinants that enable us to measure whether a pathogen is highly virulent or less virulent. In general, the most highly virulent pathogens, the ones that cause the highest mortality, death among those that get sick, tends to be lost within the population because it can't find a new host fast enough. So it ends up killing its host before it can be passed on to a new susceptible individual. So what over time, highly virulent pathogens tend to become less virulent in what we call balanced pathogenicity, which allows it to persist in the population, in its reservoir, and using us as a host. We also have ways of becoming more resistant to infection. One example of protection that persists in the population, even though it results in a genetic disease, is sickle cell anemia. So individuals with sickle cells will be immune from infection by the malarial parasite, the protozoa, called Plasmodium. And so even though this will cause, the sickle cells will cause problems in individuals as far as causing sickle cell disease and that clotting in the capillaries and such, it does afford them protection from other diseases, so it persists and it's a way that some of our bodies have adapted to resist disease, where it's especially in areas where it's endemic. But basically, in order to establish infection, there are 4 steps that every pathogen that will result in an invasion of our barriers, our mucous membranes, or our skin, will take. They'll first adhere, then colonize, then deliver effector molecules to help with the invasion of tissues. So let's look at each step in a little more detail. Adherence is a specific interaction between proteins on the surface of the pathogen, and I'm going to focus on bacteria here. So obviously viruses, they have a different way that they adhere. They have the spikes, and they are, because they're intracellular pathogens, they always have to get inside a host cell. But here I'm going to focus on these steps which apply to bacterial pathogens. So adherence is mediated by surface proteins, whether it's an adhesin protein that's on the surface of the cell or the surface of an appendage like a pili or fimbriae, which are these specialized pili that are important for attachment. Or if they don't have adhesins, they can have capsules, which also make them very sticky and easily adherent. But the adhesins form this specific interaction, so they're not able to adhere to any cell, only cells that have a receptor, which is usually a mixture of carbohydrate and protein that bind to that specific, that are recognized and bind to that specific adhesin. One example of a surface protein would be M protein, and that's expressed by Streptococcus, many streptococcal species, including Streptococcus pneumonia. And E. coli also have adhesins on their surface that help them attach to the gut epithelial cells lining the gut. And now, of course, in our gut, we have our defenses, our IgA, mucosal IgA that's secreted, that could bind and recognize the antigens on the pili, including adhesins. And then if they're bound up by antibody, of course, they wouldn't be able to interact with their cognate receptor on our tissues, on our cells. But on the other hand, many pathogens have developed virulence factors, including the secretion of enzymes that break down these antibodies, so protect themselves against the preventative effects of IgA by degrading IgA, so it can't stop them from making this attachment, this first step. They also get ahead of the production of antibody by this antigenic variation. So changing their pili, donning a new disguise, a new set of pili with a new set of slightly different proteins or adhesins, that would not be recognized by that initial wave of antibody production. And so another delayed response in developing a specific antibody response to those factors would need to occur before that part of the population of the pathogens could be eliminated from our body. So it's always this constant back and forth between the adaptive immune response as far as producing antibodies specific enough to stop this adherence and the changes that occur within the pathogen with each cycle of, with each generation. Okay, then after adherence, colonization is possible. So the bacteria really can't multiply unless they have some surface that they're attached to and some space in which to grow. They also require nutrients, so they're competing with our normal microbiota for these nutrients and have to overcome some of the toxic products that our normal microbiota would produce, as well as this ongoing innate and adaptive immune responses. And some of these ways include not just turnover of pili, but turnover of antigens on the surface. Even if the pathogen doesn't have pili, they can change the production and expression of proteins on their surface, and also to compete for nutrients by producing molecules like siderophores, which compete for iron. Those pathogens that don't have siderophores sometimes are able to gain access to iron by lysing red blood cells, which we've already seen in the laboratory. So Staphylococcus aureus, as well as streptococcal species, have hemolysins. Streptococcus pyogenes has a streptolysin, which are able to lyse the red blood cells, and then the bacteria can gain access to the iron that's stored in those cells. For those pathogens that invade, they often have a mechanism of triggering uptake by the host cells. So while adherence does not necessarily mean that the bacteria will get into the cell, but they will be able to attach to it and multiply on that tissue surface. Invasion requires other mechanisms in addition to just attaching. And in the case of gram-negative bacteria, things like Yersinia pestis, they have these type 3 secretion systems, which are essentially an assembly of proteins that act as a syringe that are present on the bacterial membrane, cell membrane, and throughout the cell wall. And then products that are produced by the bacteria are then secreted directly into the host cell across the plasma membrane of the host cell. Now, this can mediate things like the uptake of these bacteria that contribute to the invasion of the pathogen beyond the cell membrane and beyond this initial tissue that lines these barriers. And one of those examples is membrane ruffling. So we already know that penetration of the skin is quite difficult. It has to be some breach of the skin, like some cut or injury to the skin that would introduce pathogens at that site, or some vector, biting insect that would introduce the a biological vector that would introduce the pathogen directly into the blood. Mucous membranes are the most common way that pathogens enter because of the difficulty of crossing this barrier of the skin. And they can either be directly taken up or they can exploit other ways of being taken up. So a direct uptake is an example of membrane ruffling. Gram-negative bacteria that you can see here in this electron micrograph are sitting on top of the cell, will inject through this type 3 secretion system things like invasins, which will enable, even though this cell lining these mucous membranes is not a phagocytic cell, but it would allow for the uptake of pathogens through the process of endocytosis. So we'll cause this invagination of the plasma membrane. The bacteria will be taken into the cell in a vesicle. And once inside the cell, that's a really great hiding place that bacterial pathogens have to evade the immune response. Also, some bacteria secrete enzymes that will break down components of the tissue, like hyaluronic acid, also collagen, these matrix proteins that are necessary for connecting the tissue. And finally, an indirect way of penetrating and invading tissues is one that's carried out by Shigella. Shigella is this pathogen, causes shigellosis. It's a gastrointestinal infection. And the Shigella, rather than colonizing the surface of the epithelium lining the gut, like an E. coli would or a cholera would, they actually penetrate and invade that surface by being taken up by these immune cells called M cells, which are part of our mucosal associated lymphoid tissue. So Peyer's patches are these areas where you have M cells and then associated macrophage that are kind of interspersed among the epithelial cells that are lining our gut. And they would be taken in by endocytosis, delivered down below that layer of cells to macrophage. But because the Shigella can escape the macrophage before the formation of the phagolysosome, so that destructive compartment, and because they can multiply in the macrophage and then they lyse it, then they become present in high numbers in this area that's normally sterile. So there's no competition by normal microbiota here. There's no toxic effects of normal microbiota that they have to overcome. They do have to evade the immune cells there. There are still more macrophage and neutrophils that would be there. And in order to do that, they enter the epithelial cells from the basement membrane, and they take advantage of this mechanism of, I think I have it in more detail. Oh no. Where did I put that? They take advantage of this mechanism that of polymerizing the actin in the cytoskeleton, and they propel themselves from one cell to the next. So they don't actually exit the cell to infect a new cell. If they were to exit a cell, then they might be susceptible to recognition by our immune cells. And so that would speed up their elimination. But by staying within the cells of the lining the epithelium, the epithelial cells lining the gut, they're able to multiply and persist. And that process of movement is called transcytosis. So that's virulence factors that are unique to Shigella, that mechanism of movement. So basically, as far as virulence factors, they would help a pathogen to avoid antibody. So antibody might tag it for phagocytosis. It may neutralize it. In the case of viruses, it may prevent its motility if it's binding to flagellar antigens, or clump bacteria so that they can't attach or penetrate the tissues. They also want to avoid complement, which can also be an opsonin, as well as recruit immune cells of the innate immune system to that site of infection. They want to avoid phagocytic cells, or if they can't avoid phagocytic cells, they want to avoid the process of phagocytosis carried out by these cells and persist within the phagocytic cell, or prevent that fusion of the phagolysosome. So these are all ways that, and virulence factors that are associated with avoiding these host immune system mechanisms. Capsules, which will help avoid the binding of antibodies and complement. M protein, important in attachment. Enzymes like coagulase or IgA protease, important in finding ways of avoiding phagocytic cells and antibody in the case of IgA. And antigenic variation would contribute to avoiding antibody. So how does complement system come into play? Well, this is really applicable to gram-negative pathogens. So the membrane attack complex will assemble on gram-negative cells. That's the way that complement, one outcome of complement that directly kills cells by forming these pores and disrupting the cell wall and the cell membrane and the integrity of the bacteria. But some gram-negatives have a way of using mechanisms that protect our own cells, so producing proteins that will bind to the initial complement protein that starts this cascade of events, and preventing the assembly of those other complement factors, so the pore that cannot be formed on the plasma membrane of these pathogens. And so they avoid that normal killing process that may occur when complement binds directly to their surface. In the case of phagocytes, the capsule will help hide some bacteria and prevent them from being opsonized by either antibodies or by complement components. If they are bound and opsonized, and the process of this invagination occurs, some have membrane damaging toxins that can kill the phagocytic cell before the phagocytic cell forms the phagolysosome compartment. That would result in the killing of the pathogen. Whereas others will prevent the recruitment of these phagocytic cells to that area by disrupting this gradient that's formed by C5a. So remember, C5a is important for neutrophils to come into that site of infection. And by breaking up that protein, complement protein, the gradient or the signal to the immune cells is lost. Also, antibody is an important part of, I'll go on to antibody in a minute, but one way that pathogens can avoid opsonization, whether it's by C3b or by antibody, is by having a capsule. So remember, the capsule, because it's mostly polysaccharide, it doesn't make it doesn't produce a lot of good antigens that can be bound by antibodies, for example, and it would block the C3b from binding to the membrane of the gram-negative cells. It also can produce proteins that inactivate C3b before it has that effect of creating this cascade of events that would lead to the lysis of the cell. M protein, which is able to bind complement, is what inactivates the C3b and prevents it from acting. And then Fc receptors, which will flip the antibody around. So usually an antibody will bind an antigen on a bacteria and it'll be opsonized. The Fc receptors on a macrophage will bind to this red portion, the constant region, and engulf and phagocytize that bacterium. If the bacteria flip the antibody around the other way and bind it with their own Fc-like receptors, then the antibody can't function as an opsonin, has no effect on promoting phagocytosis of this bacterium. And I already talked about using that the Shigella uses this actin cytoskeleton to propel itself from cell to cell and evades the immune response by hiding out within the host tissue cells. Then some of those bacteria that are not able to escape opsonization will be taken up by the phagocyte, but they can either escape from the phagosome before the fusion of the lysosome and begin to multiply in the cytoplasm, or they can survive within the phagolysosome by producing their own factors that will inhibit the effects of the degradative enzymes. And that's in the case of the Mycobacterium tuberculosis. It doesn't multiply very much in the phagolysosome, but it's not killed by this low pH and low oxygen. And antibodies, I already mentioned most of these IgA protease changing the surface antigens, but the other one, mimicking the host molecules, is producing proteins on their surface that are not recognized by our immune response or don't elicit an immune response because they're considered self-antigens. So they're similar enough to the antigens that we see, for example, E. coli sometimes produce proteins that are similar to cell surface proteins seen on the epithelium of the gut. And by mimicking these host proteins, they evade having an active antibody response being mounted against them, at least to that antigen. So remember that pathogens are these subset of microorganisms that cause damage, and damage is what we see as far as the symptoms of a disease. And when we talk about damage, it's most commonly mediated by toxins, but there can be some microbes that, for example, they have these enzymes that lyse the cell, so those that avoid phagocytes, for example. Or in the case of viral infections or obligate intracellular parasites that are bacteria, they often lyse the cell on exit from that host cell. But basically, if the microbe is causing damage, it's usually by toxin. And the toxins can either directly be cytolytic, so cause lysis of cells, whichever cells they target, whether it's a neurotoxin or a liver toxin or other types of toxins, they can directly affect this subset of cells in our tissues, or our host immune response, if it's mounted to a large enough degree, if it's not a localized infection, if it's a systemic infection, these normal course of the immune response of fever and inflammation can actually cause damage not only to the pathogen but also to our own tissues. Even some exotoxins can elicit an overblown immune response in their mechanism of action and result in a lot of damage due to that. Basically, exotoxins are produced by either gram-positive or gram-negative bacteria. Remember, endotoxins are only found in gram-negative, so LPS is not in gram-positive bacteria. But exotoxins are those toxins that are produced and then secreted or released during bacterial cell death. They have specific effects. They're proteins, which usually means that we can make some kind of protection against them, either by using a toxoid to create a vaccine, or by using antitoxin antibodies that are against that toxin in treatment of these infections. They can act locally or systemically, but they have specific tissues that they act on because they bind first. So they have to have this specific interaction between receptors or some portion of the toxin and receptors on the tissue. So neurotransmitters are only affecting neurons. Cytolytic toxins are only lysing the membrane of a cell that they have specificity for some receptor on that cell. And they are very effective even if the bacteria infection is controlled by antibiotic treatment. The toxin can still persist and have damaging effects. And that's true of infections like staphylococcal skin syndrome, where you get the infection of the staphylococcus on your skin. The toxins result in a lot of damage and the loss of this epidermis, which can then make the patient very susceptible to other infections. But antibiotic treatment will not be enough to cure the patient. The patient will still die of toxemia if that area is not first cleaned, and in some extreme cases, antitoxin is used as treatment. So what are some examples of exotoxins? They can be specific to the nervous system, to the digestive system, or to a variety of cells in the case of cytotoxins. And they come in 3 flavors. They're AB toxins, so example of that would be the cholera toxin, where the part that the B subunit is the binding part, and the A subunit is the toxic part. So the B subunit has a specificity for example for only, as in the case of the botulinum toxin, for only neurons. And then once the A portion gets into the cell by this receptor-mediated endocytosis and it's released from that vesicle, then it has the toxic effects in stopping neurotransmission, for example, or in the case of Vibrio enterotoxin, then it gets in and it disrupts this electrolyte mechanism of electrolyte secretion and electrolyte balance, and lyses the cell lining the gut. There are membrane damaging toxins, okay, so they target the plasma membrane of the tissues that they bind. Streptolysin's also an example of a membrane damaging toxin by lysing red blood cells Clostridium difficile, which is a common nosocomial or hospital-acquired infection, C. diff, has an enterotoxin that binds and disrupts the plasma membrane of epithelial cells in the gut. And the final type is superantigens. Now, superantigens often are some of the most damaging because remember that there's a requirement for the activation of T helper cells to recognize antigen from a pathogen in combination with this major histocompatibility complex molecule. What a superantigen does is it takes the requirement for the antigen to be present out of the picture. So rather than just that subset of T cells that recognizes that antigen, okay, being activated, now what portion of the T cells are activated? Okay, so it doesn't require to have any antigen in the pocket. So would it be more T cells that are activated or less T cells that are being activated by this mechanism? Would it be many more types of T cells with many different specificities for different antigens would then be activated. They would then stimulate the B cell response and the cytotoxic T cells. And so you get a lot more damage from this really overblown immune response due to these superantigens. So 1 in 5 T helper cells are being activated rather than something like 1 in several thousand or hundreds of thousands. And one example of a superantigen toxin is the Staphylococcus toxic shock syndrome toxin, as well as the Streptococcus pyogenes, this erythrogenic toxin. And Staphylococcus enterotoxin is another example. So what about endotoxin? We already talked about this, this component of the LPS that's responsible for septicemia. Septicemia is characterized by the symptoms of fever and chills, a rapid drop in blood pressure, and remember that antibiotics, even if the cell of the bacterium is disrupted, the lipid A still is intact and has its toxic effects. So while the bacteria may not be multiplying or producing more bacteria, the toxin is still present. And we don't have any antibody production against the lipid, unlike exotoxins, which are proteins which make really good antibody responses. And the toxins can be neutralized as part of our normal immune response just by the binding of the antibodies to those toxins. Remember, they can be neutralized similarly to the way that a virus can be neutralized by antibody. So what happens is this septic or endotoxic shock. And in the case of gram-negative septicemia in the hospital setting, it's most commonly acquired by IV use. So the introduction of a bacteria, not necessarily. So in the hospital there are many protective steps that are taken to be sure that the fluid that is being administered by IV is free of lipopolysaccharide. Okay, so remember, it's heat stable. Unlike other toxins, the exotoxins, they would just be completely destroyed by autoclaving. No need to worry about those fluids that carry exotoxins. But in the case of endotoxin, it's heat stable, so it's not destroyed by autoclaving. So you have to filter it out and to be sure that these IV fluids are free of lipid A. And it's really the lipid A effects on our immune system that cause the drop in blood pressure. So activation, the lipid A is an important pyrogen, and also inducing fever, and also it would stimulate cytokine production by macrophage that are part of our normal innate immune response against these infections, result in the blood vessel dilation, also that's associated with the drop in blood pressure. So this lipid A would stimulate several effects that contribute to septicemia. And finally, avoiding immune responses by viruses. So there are mechanisms that viruses have, as I mentioned. They can avoid antibody. So outside of the cell, they're inert, but they need to be able to find that new host cell and bind to it. So antibody can effectively neutralize them by binding up their spikes, right? But in the case of some viruses, they're able to bind antibody in that constant region. So they, rather than getting bound up by the antigen, I don't have a pen. There's one. So rather than getting bound up by those two arms of the antibody here, they bind down here and they are actually taken up by receptor-mediated endocytosis into a cell and then can reproduce in that cell by that mechanism. They hide out from the effects of the neutralizing effects of the antibody in that way. Also, they have products that they can secrete that will lyse the plasma membrane when they leave, so they're damaging cells in that way. They can make their own version of class 1 MHC and put it on the surface without any of the viral antigens so that the T cells won't recognize it as being a virally infected cell. Or some of them stop the protein production and expression by the host cell so that no MHC is on the surface. Of course, we do have natural killer cells that recognize a cell without any MHC and will destroy it. So it kind of buys the virus enough time to replicate, but it doesn't completely hide it from our immune response. But when there's a fake MHC molecule without any of these little green spots, which are viral antigens present, then it is effective at preventing cell death of that infected cell by either cytotoxic T cells or by NK cells. All right, so finally, they can form these large groups of cells where they can go, it's a fusion of cells so that they don't need to exit the host cell in order to infect a new cell. They're called syncytia. And I already mentioned they can attach the Fc portion of the antibody to avoid that. So are there any questions about the pathogenesis? Okay, so remember, a week from today, the study guide is up for exam 3. And if you hold on for a minute, I will hand back your